Alright, so today is day two, or this is a review of day two of nucleic acids, and specifically looking at DNA a little bit more so than RNA. But I wanted to go ahead and talk about some of the important parts about analysis of DNA, and so we are going to go ahead and begin doing that on the next, or in just one moment, so hang tight. Now there are a couple of things whenever it comes to any sort of DNA or RNA molecule. What you want to be familiar with is the bond types. So DNA and RNA have a couple of things in common. We previously talked about the different nitrogenous bases that are common for both DNA and RNA, but what I wanted to talk about first of all was the bonds. So whenever it comes to forming a DNA double helix or a DS DNA molecule, the bonds that hold, there are basically two significant types of bonds. So two significant bond types. They are phosphodiester bonds, and those are basically a phosphate molecule, phosphodiester bonds, a phosphate molecule that links sugar to sugar, to sugar to sugar to sugar to sugar, and so on. Additionally, hydrogen bonds hold basically nitrogenous bases together with one another. It's important to know that the dsDNA, the double-stranded DNA, that is a double helix. And at the interior of that helix, of that double helix, those are your nitrogenous bases. On the exterior, that's where you're going to find those phosphodiester linkages. But to put it very crudely, this is basically one side of a backbone of a DNA molecule. You're going to start on our 5' end, and what we have here is a phosphate that, let's see, H2. I'm going to draw the sugar pretty succinctly. And then on our 3' end, we have another phosphate. Let me go ahead and complete these. Negative, negative, well, negative. We're going to have that there, and then CH2. Again, the sugar molecule. O, P, O. I'm just going to put an R group there. Basically, your phosphodiester linkage is more or less right here. So we have one phosphate group that is connecting the 3' carbon of one sugar molecule to the 5' carbon of the next. And this continues on. The 3' hooks up to the 5'. If we look over here, our base for this strand and our base for this strand, what we would actually have is a complementary piece of DNA, in which case we have the complementary, right, C base, complementary base, hydrogen bonds, and our complementary base over here, hydrogen bonds. So A would bind to T, and G would bind to C, and so on. So those are, and what's important about these bases is these are hydrogen bonds that stabilize these. So hydrogen bonds here. So when it comes to, it's pretty commonplace for some people to know that a DNA molecule is negatively charged, but where that actually comes from is something that requires a little bit more insight into what the structure of that molecule is. And here you go. There's your negative charge. There's another negative charge. And on the opposite strand, you're going to see those same negative charges. So it's the phosphate groups that give this molecule an overall negative charge. So when it comes to thinking about a DNA molecule, a dsDNA molecule, one of the things that's quite common is to know the base pairing. So you would know that adenine binds with thymine, cytosine with guanine, adenine with thymine, cytosine with guanine, everything like that. And, guanine is going to bind with cytosine. One thing that's important to recognize about this, and in addition to knowing that they're hydrogen bond stabilized, is to know something about those base pairs. So AT, how many hydrogen bonds are there? There are 2 hydrogen bonds that exist between these 2 base pairs. Whereas CG and GC, there are 3 hydrogen bonds. So when it comes to looking at those numbers of hydrogen bonds, this is pretty significant, and it's pretty neat when it comes to thinking about something on the genomic level. Now, there are organisms that live in extreme environments, in which case their genomic concentration or their abundance of GC content is higher than their AT content. And the reason for that is because, well, there are 3 hydrogen bonds between each G and C and G, compared to the 2 hydrogen bonds between A and T, which means if you have a higher number of GCs in your genome, you have a higher number of hydrogen bonds within your genome. So it's something that gives the molecule actual structural integrity. But one thing that I like to ask a question about relating to these is I like to ask a question that would ask something like, a newfound organism is 20% A. And then you're prompted with a couple of different options, like explain, elaborate. What else do you know about this? Well, one thing that you know is that if an organism is 20% A, it is also 20% T. That total accounts for 40% of the genomic, nitrogen bases of that organism. The other remaining amount is 60%. Split up among 2 different base pairs, or 2 different bases, C and G. And since this is 60%, you can break this down to 30% C and 30% G. Now this is known as, and this is an important part of this, is known as Chargaff's rule. Ultimately, this is very simple math, but it's something that, a good number of people make a mistake. They look at, well, it's 20% A, therefore it must be, I don't know, that means it's, I don't know exactly how to, I don't want to say anything that will throw you all off or anything like that, but this is, just think about what matches up with what and what remains after that. The last thing that I wanted to talk about or to kind of reflect upon this day's lecture is restriction enzymes. And these are enzymes that carry out one specific function. And what they do is they recognize DNA or they see DNA and they recognize a specific sequence. If we take, for example, EcoR1. EcoR1 is an enzyme that will see a double-stranded piece of DNA. This is our double-stranded piece of DNA. And EcoR1 will jump on this piece of DNA and scan. And what it is going to be looking for is a specific sequence. It's going to look for GAATTC. And there are tons of different restriction enzymes, and they all have unique sequences, or they have unique sequences. Just as EcoR1 is going to look for this, well, we know that this double-stranded DNA has a complementary strand. So it's going to have GAATTC on the other strand. And what this enzyme is going to do is it's going to break a pair of phosphodiester bonds between that G and that A. And whenever it does that, well, you have this kind of fracture right there. This is going to break a phosphodiester bond, phosphodiester. So it's going to break these phosphodiester bonds, and in time, Well, the only bonds that are left are these hydrogen bonds between each of the nitrogenous base pairs, or the nitrogenous bases. So the result is going to be, well, pieces of DNA that have complementary regions but are no longer attached to one another. This would be one of them, and this would be your second one. It's going to look like G, C, T, T, A, A. Those two pieces of DNA are going to result from EcoR1 breaking a pair of phosphodiester linkages. So restriction enzymes, they will scan double-stranded DNA and they will find their recognition sequence, they will find what they are looking for, and they will break your phosphodiester linkage. In some cases, you'll end up with products that look like this, where there are these overhangs. These are referred to as sticky ends. There are other restriction enzymes that will see a double-stranded piece of DNA, and what they will do is they'll say, let's just, for the sake of simplicity, CA, GT. What they will do is they will break a phosphodiester linkage there and there. And whenever they do that, what they're going to be doing is they're only breaking, well, they're breaking two phosphodiester linkages, but they're not breaking any hydrogen bonds. And actually, what I know about, or what you should know, is that these sequences are going to be reverse complements. So that's not a good example. A good example would be something like this. AC is going to break this phosphodiester linkage here and here. What it is going to do is it's going to see a sequence here that it likes and a sequence here that it likes. These are palindromes of one another. So at any rate, if a restriction enzyme breaks a product like that, there, it's going to result in two products that look like this, ATCG plus GT CA. No hydrogen bonds are going to be broken, and instead these are going to be different from our previous product. These are known as blunt ends. So those are some pretty important things about restriction enzymes. One last thing that I want to talk to you about restriction enzymes, however, was the frequency of seeing the products. So if we think about a piece of DNA, if we think about a randomly generated piece of DNA, you have to think about probabilities, or it's important to think about probabilities when you're thinking about restriction enzymes. A restriction enzyme is going to look for a piece of DNA, or it's going to look for a specific, at least I'm going to say 4 base pairs long. And it has a specific sequence that it's looking for. And let's say it's looking for this ACGT. A question that I like to ask is, in a randomly generated piece of DNA, how often will this sequence be found? Okay, to answer that question, you have to do a little bit of math, and it's not very complicated, but you have to think about each of these positions. So what are the chances that when you randomly generate a piece of DNA that you're going to put an A? Well, 1 out of 4. Okay, next, the C. What are the chances that a C would end up in that next position? It's still 1 out of 4. G, still 1 out of 4. And T, 1 out of 4. So what you're going to do when you look at all these numbers is you're going to multiply them all out. So what are the chances that you would actually generate ACGT? Well, 1 out of 4 times 1 out of 4 times 1 out of 4 times 1 out of 4. Which would ultimately be 1 out of 256. So a question that I like to ask on a regular basis is, if you randomly generated a piece of DNA that was, let's say, 700 base pairs long, what are the chances that you will find ACGT one time. Well, they're pretty good, because if you have a piece of DNA that's 700 base pairs long, well, you're probably going to find ACGT once every 256 base pairs. And so a good question would be, how many times would that randomly generated piece of DNA be cut by the enzyme that recognizes ACGT? The answer to that would probably be 2. You can make an argument for 3, but at the same time, these are all probabilities. We're going to go into restriction enzyme cutting patterns in the next segment. But with that said, I hope this was helpful. Thank you very much for watching. Best of luck. Thank you very much.